Welcome to another edition of Cannabis Conversations. I'm Martin Lee with Project CBD, and today we're going to be speaking with Matt Elmes. Matt is the Director of New Product Development at CannaCraft, a major cannabis company in California. Matt heads up the science team there, and CannaCraft has been a supporter of Project CBD for several years. Matt is also holds a Ph.D. in molecular and cellular biology from the State University of New York at Stony Brook, where he also did his postdoc work. And much of that work, I think, focused on the endocannabinoid system. And that's what we're going to talk about today, and just sort of roll back the clock for a moment. To the 1990s, the decade of the brain, there were several discoveries about the endocannabinoid system, and it identified certain components that are comprise what scientists would refer to now as the canonical cannabinoid system, the receptors in the brain, the CB1, CB2 receptors, the cannabinoid receptors in the brain and body, also the endocannabinoids that bind to these receptors, and then there was identification of certain proteins that were involved in the biosynthesis and the breakdown of these endocannabinoids. Well, your group at Stony Brook, Matt, added a whole nother dimension to understanding the endocannabinoid system. You were involved in the discovery of what's known as transport molecules. Maybe you could explain what a transport molecule is and what it has to do with the endocannabinoid system.
Sure. So the transport molecules we're talking about are the fatty acid binding proteins, or I'll call them the FABPs. So, well, first they have a canonical role in transporting fatty acids throughout the body, and that's why they got their name. However, my group at Stony Brook, particularly my mentors Dale Deutsch and Martin Kachoa, led the discovery that these molecules are also transporting the endocannabinoids, namely that's 2-AG, 2-arachidonoglycerol, or anandamide.
And why would they need to be transported? Why don't they just sort of go in the blood, and, wherever they need to go?
That's because endocannabinoids and cannabinoids in general are highly lipophilic. So they're like oil and water. They will sequester to the oil phase and stay away from water. Now remember, all the cells in our body are essentially a bubble of oil with water on the inside. That's the cytoplasm. So we knew the endocannabinoids are made on the outside of the cell and they'll stick to that lipid layer. However, the enzyme that breaks them down, particularly we're talking about the anandamide breakdown enzyme, which is known as fatty acid amide hydrolase, or FA, is found on the endoplasmic reticulum.
And so that's inside the cell.
Yep. So it's essentially in the middle of that bubble of oil. We knew the endocannabinoids somehow got from the outside to the inside, but no one ever knew what that process actually was and how that happened. There were a lot of theories out there. There was vesicle diffusion or protein mediated. So about 10 years ago, our group found that the FABPs in the brain, there's a few specific isoforms that are found in the brain, essentially pick up those endocannabinoids from the outside of the cell, carry them inside the cell, and pass it off to that enzyme, or other areas too, because these are also interacting with nuclear receptors, such as the PPAR receptors. So they're essentially just shuttle proteins. They're picking up cannabinoids and transporting all throughout the cell.
So these transport molecules, these fatty acid binding proteins you refer to, what are the phytocannabinoids, CBD and THC, if anything? What do they have to do with this?
So it's actually where my whole Ph.D. project came in at Stony Brook. So as I mentioned, my mentor had discovered this class of proteins being responsible for endocannabinoid transport. So phytocannabinoids are sort of a similar size, and they're also very lipophilic. So they have to overcome that same barrier of how to cross these aqueous layers in the body. So I focused on, hey, maybe FABPs are also transporting THC and CBD or other phytocannabinoids. So we did some testing, and I found that in vitro, these things are actually binding. They have an affinity for THC and CBD. And then we kind of did that a step further and showed that this is actually happening in the body. We can compete, THC or CBD can compete for anandamide uptake.
What does that mean, though? What are the implications of that? So if the endogenous cannabinoids, like anandamide and 2-AG you mentioned, they're hopping on these transport molecules and getting through the cell and doing their thing, what does it mean then that CBD and THC could do the same thing on the same fatty acid binding proteins?
Sure, I think there's potential that they are modulating the endocannabinoid tone in their own right. Because again, as I mentioned, that FABPs are picking up the endocannabinoid anandamide to be broken down. So if we block that transport step, there's going to be less anandamide getting broken down, hence more anandamide in your body that's able to signal and hits cannabinoid receptors.
And what would be the implication of that, for example, of an enhanced signaling from the endocannabinoids on
Exactly.
these
So
receptors?
now if we have CBD, say, competing with anandamide for that binding to that protein, less of that anandamide is going to be able to bind and get broken down. So the fact that CBD is around and present might actually lead to increased levels of anandamide and increased endocannabinoid signaling.
So that would have potentially significant health benefits.
Certainly. So this is all very new work, and we are still learning more about what this is doing and how this is interacting between the phytocannabinoid and the endocannabinoid systems. But I certainly think there's potential, strong potential, for CBD, a partial mechanism of action for CBD to be modulating the endocannabinoid tone by decreasing the rate of anandamide breakdown in the brain.
So in essence, the implication would be that CBD kind of boosts our own endocannabinoid signaling, which might be sort of like a natural high, you might say. Yeah.
Yes, yeah. Again, we are still working some of the pieces out, and we can't definitively say this for sure, but I think it's certainly a partial mechanism of action.
So one of the knocks on cannabis use medicinally is that, okay, you smoke a joint or you eat an edible, you swallow a gel cap or something, take a tincture, and it has a global effect. It affects receptors all over the body and brain. Whereas if you've got a problem in one place, what do you need all the rest of it going on, you know? So what about the fatty acid binding proteins? Is it still the same problem here? If CBD or THC or some other, maybe a synthetic ligand was targeting these fatty acid binding proteins, would you still have the same global effect, and would that be a detriment as far as a pharmaceutical company or pharmaceutical development was concerned?
That's a great point, Martin. And FA inhibitors have been developed in the past. Now, again, that's the enzyme that breaks down anandamide.
So that would also be kind of a global thing, if it would
Exactly.
break down everywhere.
Because the FA you find in the brain is also the FA in the liver, is the FA in the kidney, is the FA everywhere in the body. Now, the FABPs are 10 different isoforms found in mammals. So in humans, there's 10 different types of FABPs, and they have tissue-specific expression patterns. So the one you find in the brain, it might not be expressed in the liver or any other part of the body. So what our group actually started doing, and we're in the process of doing, is developing these endocannabinoid transport inhibitor compounds. And we're still in the preclinical drug development phase, but pushing it along. So we're trying to target just the FABP isoforms that are found in the peripheral nervous system and in the brain. So that would have anti-inflammatory and anti-pain effects by modulating our own endocannabinoid system.
But also a very kind of specific effect, because you could, if there are these different FABPs in different parts of the body and the brain, by hitting one of them and not the others, you could have a very specific effect, unlike...
That's exactly right. So that's what we're trying to target, just those few isoforms that are found in the brain. So we can have this anti-pain effect without shutting down our FA enzyme in the liver, where we need that enzyme to process other vital functions in the body. So it gives us a little bit of more finesse and a precise way of targeting just where you want it to be targeted, just to modulate the endocannabinoid system and these specific parts of the body.
And that's something I would presume pharmaceutical companies might be interested in as part of their drug development efforts.
Certainly. So we had some IP around this whole idea and some of our early chemical compounds at Stony Brook. A company called Artello Biosciences, it's a small biotech company, had licensed these patents from us. And they actually had funded my postdoctoral work doing some preclinical drug development toward these endocannabinoid modulating drugs. So there's certainly interest, and we're trying to push this into phase 1 clinical trials to learn more about how these are working.
So last question, what is it like then to go from this setting in academia where you're working at a high-powered lab doing sort of this hard science, or maybe you've got these applications now with beginning or hopefully in some kind of testing program for drug development. What is it like going from that environment into the cannabis industry, sort of the heart of it in California at CannaCraft, where you're focusing presumably on product development, but different sort of work? What's been the transition like for you?
Well, I don't want to say the stress goes away. It's certainly a lot of that, but the work's very interesting. The pay is certainly better, I'll say that much. So I think my transition might have been a bit easier because I'm moving from sort of a hard science lab into an R&D environment where your kind of scientific knowledge is still valued and we're still doing experiments and things like that. But there's less of a focus on obviously grant writing and where to find that next the grant money to keep funding your research and funding your own payroll. Now it's more about making the best quality product we possibly can, and ultimately money, of course. But the goals are a bit different, and it's more of a typical 8:30 to 5:30 weekdays, whereas in my old lab I was just weekends, nights. You never stop thinking about it. So you can kind of turn it off when it's more of a 9 to 5 type of job. But so far, I'm loving it, and it's been great.
Great. Well, thank you very much, Matt, for sharing.
Thank you, Martin.
Well, thanks very much, Matt, and it's been another edition of Cannabis Conversations.